Ok, bonjour tout le monde. On va commencer tranquillement en laissant les gens se joindre à nous. So welcome to this seminar, joint seminar between our institute, IBIS, and the training program on the evolution of fungal pathogens that was just created this fall. Today, we are very happy to welcome Maitreya Dunham. Maitreya is a professor at the University of Washington in Seattle. And I still remember the first time I met Maitreya. It was at the Cold Spring Harbor Biology of Genome meeting. I was presenting my poster, my first work on yeast. And I was the only one working on yeast in my lab at the time. And it's a good thing I didn't know who she was when I presented her my poster, because I would have been way too stressed to make a good impression. Because Maitreya already back then, I don't know when it was, probably 2004 to 2005, she was already doing pretty impressive work on yeast. So Maitreya did her bachelor's degree at MIT in biology, and then a PhD at Stanford. And when I met her the first time, she was a fellow at Princeton, where she had an independent position, kind of a super postdoc that had their own lab and had their own team and funding, and they could really innovate in terms of research. And Maitreya really was one of the first people to do experimental evolution with yeast, one of the first people to study genome evolution at the time using DNA microarrays to look at gene duplication, segmental duplication, aneuploidies. So a lot of things that we still do now, she was already doing almost 20 years ago. And she is really like at the forefront of of the exciting research that people do using yeast. And now she extended the work that she did in yeast to also use yeast to study the function and the impact of mutations on human proteins. So she's made many important contributions to science. A lot of it using functional genomics and experimental adaptation. So she studied, for instance, the role of aneuploidy in adaptation. A lot of her work is on natural variation within and between species, and trying to understand the impact of this variation at the molecular level. She's done some work also on the genomics of domestication in yeast, and a lot of cool work on the evolution of genome organization. Some of people in our lab have funded companies that are now using the tools that they developed together to better do genome assemblies. Some of you may know about this. So Maitreya is always very highly stimulating person to talk with. We just discussed like for 5 minutes, and we had already cool ideas about things we could collaborate on. And so I was very excited when I saw the paper that they published on bioRxiv, where they did experiments with high school students, so they could learn about evolution using experimental evolution, but at the same time do something discovery experiments where they discover new mutations and chromosomal changes that are associated with antifungal resistance. So in the framework of the training program, I thought it would be a great idea to have her as one of our first speakers, where they can, so she can tell us about how you can put these experiments together and get high school students to contribute to doing exciting science. So thanks a lot for being here, Maitreya. We'll have you here physically eventually, but I think Zoom will do for the moment. And thanks again for your time and for meeting with people after the conference.
Wow, what an introduction. Thanks so much. And thanks so much for the invitation. It's actually really perfect. The timing on this could not be better. We're in the middle of working on the revisions for resubmitting this paper, and talking to this audience is exactly who I'd like to get some feedback on, because while the experimental evolution side of things and the genomics side of ththings is things that we have a lot of experience with, I'm trying to apply this to antifungal resistance. There's of course a huge literature about that and lots of knowledge about that, and I am willing to bet that we've overlooked some cool things in our own data. And so I'd encourage you to take a peek at our preprint. I've got a link to it later in the talk. And also, one of the funny things about Zoom seminars is that if people want to meet with me later, that's fine. Like I could meet with you next week or some other time. So if after the talk you're like, you know what, I wish I'd gotten on your calendar, then just feel free to email me and we can set something up. So today I'm really excited to talk about how we have taken our kind of standard evolution regime where we take populations of yeast, we subject them to new environments, and allow them to evolve for a few months. And then we sequence their genomes and see what happened and what did it take to invent these new capabilities. So this has been a kind of the model for lots of experiments in my lab with variations and all sorts of the strategies here. So for example, this is just showing transfer between flasks as a way of doing evolution, but we very typically use continuous culture gadgets, for example. But the path is definitely more or less the same. Different environments, see what they do. And of course, the sorry, my pointer has stopped. The goal, of course, is to understand evolution, and being able to watch evolution happen is a great way to do this. We can have the entire evolutionary history of these populations in our freezer. And the idea is to understand evolution of yeast, which is important for things that yeast do, like domestication and all the different fermentation properties that they have. But also, we think that this is somewhat of a model for things like how do cancer cells evolve and how does antibiotic resistance evolve. So the other piece of the puzzle today that came together to make today's talk is one of these moments of luck in science where it's really hard to have set this up on purpose. And so this is now saying, can we take experimental evolution into the classroom? And it all started when Ryan here, who's a teacher in Southern California at the Westridge School for Girls, wondered if he could recapitulate the Rich Lenski's long-term evolution experiment project with E. coli, but in his high school classroom. He had been teaching AP biology for a few years and was kind of unsatisfied with how they presented experiments and research and lab work. And he wanted to see if he could turn his classroom into a research lab and engage students in research and see if that could be a better way to teach some of these science concepts that he was working on. And while he had come across Lenski's experiment, it turns out that the AP biology standards are much more about learning about eukaryotes. And so instead of emailing Rich, he Googled yeast experimental evolution to see if there is anybody out there working on this. And funny enough, I have a book chapter that I wrote several years ago, which I think it's cited twice or something, and maybe both of those are by me, I don't even know. But it has very high Google rank. And so he clicked on it and he sent me an email saying, hey, would you be interested in doing experimental evolution with high school students? And the match was made. And the other person that made this all work is my postdoc, Bryce Taylor. And Bryce came into my lab with yeast genetics experience from his graduate work with Ian Ehrenreich, but also a love of education and outreach. And so his goal was to try to find a position in an undergraduate oriented university where he could run a lab run by undergraduates. And so this actually ended up being perfect. And he just started that job exactly a couple of months ago. He's now at Loras College, where he's going to continue on some of this work, and my lab will maintain a franchise of it essentially. And so what's been great about this is that experimental evolution of yeast actually matches so many of the standards of things that Ryan wanted to teach in his class. And that, obviously evolution and genetics, but also because of how the students get to think about how mutations work and where the mutations are having their consequences, they ended up also learning a lot of cell biology, metabolism, and molecular biology along the way. Okay, so how did we put this all together? So I'm going to step through different components of it and then show you some of the data that we got from the students. So first of all, we had a few goals for this, right? One was this idea of an authentic research experience. So we wanted to come up with something that was, it would work, it would be satisfying, but at the same time, there would be opportunities for discovering new things. All the answers wouldn't just be there for the students to look up. We wanted to use evolution as this connective concept to learn about kind of all sorts of different levels and layers of biological organization, which is really important to the next generation science standards in the United States. And we wanted the students to get to experience lots of different aspects of an authentic research experience, including having some agency in their own experiments, as opposed to kind of what are sadly typical teaching labs, which are very cookbook-like. We wanted them to be able to collaborate with each other, and we wanted there to be opportunities for competition as well. So what we came up with is a series of modules, and we're calling this lab YEVO. And Ryan has used the entire lab to scaffold essentially his entire year's curriculum. And we have other teachers now that we've also started to work with who have just done a couple of these modules or are actually working on new modules. I have a teacher in Seattle who's working on a genetics module with us for this school year. And so the basic concept is that we have the students do experimental evolution via serial transfer. And the experiments that I'll tell you about today are for evolution of drug resistance, but I think you will quickly see that this is a model that is really infinitely adaptable. They then send their evolved strains to us and we sequence them. We return the results to the students. This is actually something that I'd love to brainstorm about with people. We have not yet been able to come up with a really great solution to having the students participate in the sequencing and the raw data analysis. Although one of my other teachers outside of Seattle in Issaquah actually just got their hands on a MySeq. So we're hopefully going to figure it out with them. But this has been something where we kind of almost act as a core facility. And we give them back a curated set of mutation calls, and then they are the ones that have to go through those and figure out which genes do they think matter. Then we have them measure phenotypes of their evolved strains by looking at, say, minimum inhibitory concentration for the drugs. And they also do competition experiments to measure fitness. And then they can measure other phenotypes that are unrelated to the trait. So not exactly drug resistance, but how do they, say, grow in other environments? And that leads them to discussions of more nuanced aspects of of evolutionary biology, such as trade-offs. Okay, so the first thing we needed to do, like I said, my lab does a lot of experimental evolution, but in continuous culture. And so the chemostats that we've developed, while we have simplified them and miniaturized them over the years so that we can do lots of chemostat experiments relatively easily, this is not something I would recommend taking into a high school classroom. So instead, we went with the serial dilution technique. And we also tried to figure out lots of ways to kind of decrease the probability of contamination and things like that throughout. And so we came up with using individually wrapped sterile cotton swabs to do the transfers so that the students aren't using pipettes or anything else that where bad technique could possibly doom the experiment. And all they do is they have their cultures going. And the nice thing about yeast is they can just sit on the bench at room temperature and they're perfectly happy this way. Although I should mention one of Ryan's classrooms, they did have one roller drum in an incubator, a 30-degree incubator. And the students actually figured out that the yeast grew better in the roller drum in the incubator. And they started coming to class early so that they could get a spot in the roller drum and their colleagues who came later would have to leave their tubes on the bench. They figured that out themselves. Anyway, so what they do is they do a transfer and then they make some notes about how well the cells grew and what dosage they were working at and things like that. And the way that this kind of goes onto the calendar for the class is really variable. It really depends on the classroom. So in Ryan's class, they meet every other day. And so they were doing transfers every 2 to 4 days because there's weekends in there as well. And then some of our other classrooms have done a weekly transfer schedule. So they only do a transfer once a week. And remarkably, both of those strategies work. We have seen some minor differences in mutation patterns, but we're not really sure if that is because of the differences in timing or what. Okay, so the other component of this is what ancestor strain are we going to start with? And so we're using a lab strain for all of this. But we decided to use these really pretty strains that we got from Jeff Buca's lab and others that the students at the Cold Spring Harbor yeast course had been making that express the beta carotene biosynthetic pathway. We affectionately call these ones the carrot yeasts. And so these are all expressing, all sorts of different genes from like coral and bacteria to make these pigments. And so that leads to a little bit of a discussion of kind of like synthetic biology topics, which the students really enjoy. And the student team gets to choose one of these strains as their ancestor, which kind of gives them almost like a team identity. Although I have to say, in our surveys that we did about what students liked and didn't like about the class, one of the teams that got this gray strain was not happy about it. They complained about it in like multiple points of their survey. So, but we need the multiple colors so that we can do another part of this, which is doing competition experiments. So if the students are all evolving different colors, then we can compete them and see who has evolved the best yeast at the end of the experiment. It turns out that the color is also really useful to check for contamination. Since multiple student teams use the same bench over the course of a school day, we did occasionally have some accidental cross inoculations, I should say. They weren't contamination. We had shockingly few contamination events, actually, but you could tell when the culture turned muddy, it was like, uh-oh, something's wrong with this. And so luckily, we keep the samples in the fridge after they've been used so that in case of disaster, we can kind of go back in time and start again. Okay, what's next? The next thing we need, of course, is a selective condition. So Ryan was able to come visit the lab for a couple of weeks in the first summer that we were devising this. And he and Bryce went to the drugstore that's just down the street from the university, and they bought every over-the-counter antifungal, which apparently the cashier took their money very gingerly, I'm told. And then they tried all of these different antifungals against laboratory strains of Saccharomyces cerevisiae to find out which ones would work well and which ones were easy to work with. So some like didn't dissolve very well in our media. Others, the yeast didn't seem to care about. And the one that was the winner, though, was this one here called Fungicure. This comes in a spray bottle to spray on your toenails. And the active ingredient is clotrimazole. And so that was really great because of 1 to 50,000 dilution of this kills lab yeast. And this is something that's sold over the counter. So we reasoned that it would be pretty safe for the students to work with, especially at such a high dilution. And of course, azoles are such an important class of antifungals. And back to one of our goals, which was that there needed to be enough background for them to really ground their results and frame them in previous information, but also some new aspects. The mechanism of action of this is known. It inhibits ergosterol biosynthesis, and there's a whole bunch of resistance mutations that are known from both pathogens and also work in laboratory evolution experiments, in fact, with things like Candida albicans and screens and cerevisiae and things like that. The aspect of this that we really liked was that you start with a 1 in 50,000 dilution, but the students get to decide when they think their yeast are doing well enough that they're going to up their dosage. And this led to all sorts of fun student interactions. So one of the things that we would watch them do was, like I said, they were sitting at the same desks, right, in different periods, and they were like snooping on each other's notebooks to see how high other students had gone. And some of them would be like, oh my gosh, the other people are 10-fold higher than us. We got to go, go, go. And other people had a plan. They were like, we're just going to increase this slow and steady, and we're sticking with the plan. And this has become one of my favorite questions to ask on like qualifying exams for students doing evolution is, what strategy would you propose to the high school students in order to kind of evolve the most resistant yeast for these experiments? This also leads to the possibility of failure, which I think is important for really truly having agency here, right? The students can go too far too fast. We've had some extinction events happen. And so when that happens, we go back to the fridge where we've kept some of the backup tubes, and they're able to keep going with those. So that's been their learning experiences, but backed up so that it doesn't stop their experiments entirely. We also have let the students work in usually 2 to 3 replicate cultures so that they can kind of try different things with different tubes if they want to. Okay, now to get to the data sets that I'm going to talk about. So one other nice thing that happened was we were able to recruit a new partner on this, who's Paul Rowley. Paul is a yeast geneticist at University of Idaho. And Paul and I wrote an NSF grant together and put this teaching lab in as part of that. And the teaching lab part has actually worked fantastically well. The science aspects of the grant have ended up being a little bit harder. We were wrong about one of our hypotheses, it turned out, but the teaching lab was able to get Paul on board, and also he was able to recruit 2 teachers at a local high school to him, and this is a public high school. Westridge is a private school, and that's why Ryan was able to come and visit us for 2 weeks, and some of the other nice resources that they have available. So we're kind of calling this a private-public partnership, where it really did take a highly resourced school and a PhD level teacher. Ryan has a PhD. In order to start developing a lot of these modules and to work closely with us on figuring out what high schools need. But we've now developed the modules well enough that we think that any school can use them, although certainly differences in resource levels have mattered. So for example, one of the public schools we've been working with outside of Seattle in Issaquah, they were like, oh great, we'd love to run this lab. We've got all these test tubes. What else do we need? Do we need lids for the test tubes? We don't have any lids. So we had to like get them lids in order for them to be able to run the lab. So, you know, it really is a communication with the teachers that you're working with to see what they have access to. And the other expensive component of this, of course, is the genome sequencing. And so because we're able to use the genome sequencing for research goals, we've been able to fund that off of the research aspects of various grants. And justify it that way, because the genome sequencing here is completely out of the range of most high school science budgets. So, okay, so the Westridge School first did their experiment for 7 weeks. The plan was to do the evolution experiments in the fall. They would send us the strains over winter break. We would get them sequenced, and then we'd send back mutations once the students came back in January. And I knew we were onto something, that the students were really enjoying. This is one of their actual notebooks, that they were really enjoying the yeast life when after they came back from winter break, they asked if they could keep going with their evolution experiments. I was like, oh my goodness, we're onto something. So they ended up evolving over the entire year. So we took this 7-week time point and they did a lot of work with that, but they kept going and we ended up sequencing some of their later time points as well. The Moscow high school classes, they did about 12 weeks, and these were these weekly transfers. And we have 2 years of data from Westridge and in the data set that we analyzed here, and we have multiple classes worth of students. So it's 3 teachers total and something like 6 or 7 classrooms, I think. So what we did was they sent us their populations and we isolated clones from them and then retested their drug resistance phenotypes just to make sure that we were sequencing a strain that actually did have enhanced drug resistance. And so this is just a graph of the minimum inhibitory concentration of the ancestor strains are pretty low, but in all of the 2 high schools, the early time points, we saw a range, but mostly improving. And then in the final time points from Westridge, these 8-month ones, we saw an improvement even more. So a lot of these strains, actually, we think that they ended up selecting the clotrimazole is dissolved in ethanol. And so we think they were actually like having problems where they were so high in concentration that this might not have actually been the experiment they thought they were doing at the end. But anyway, these are pretty good resistance phenotypes here. Okay, so we sequenced them, and the sequencing was done by my lab, by Bryce, and also we had the great luck of, I was giving a talk about this in some other venue where Fritz Roth was there, and he said, you know what, I teach a genomics class to undergraduates at University of Toronto where they sequence something and then they do all the data analysis for it. How about if you send me some of your strains and we can do the sequencing for you? And so Atina Cole and Fritz used their class to do some of the sequencing for us, which has been really helpful. And we're hoping that this will eventually lead to kind of a way that the undergraduates can communicate with the high school students whose experiments they were analyzing, so that the high school students can have some, discussion of like, what's college like, and things like that that could lead to more a better educational experience all around, and also allow students to imagine their possible futures in science and taking science classes in college. All of the strains had at least one obviously causative drug resistance mutation. But the strains were really varied in terms of what their genomes looked like. So all the strains contained point mutations, but we also got a lot of strains that contained copy number changes, transposon insertions, and also changes to organelle genomes. So this was great because it let the students really think about all types of different possible sources of mutation and how those might affect these phenotypes. And here's the link for the paper. If you just search for Taylor and Dunham, you'll find it. And there's also a link from my website. Okay, so I'm just going to step through some of the mutations we found. I decided not to go exhaustively through all of them, just because for the sake of time, and I want to leave some time for discussion at the end as well. But I want to get some ideas about some of these as well. So we found a lot of copy number changes, and these are known to be important for how pathogens evolve to evolve drug resistance. And one of those is amplifications that include ERG11. So we saw 38 strains that had whole chromosome 8 amplifications. And then we saw a bunch of strains that had shorter segmental amplifications. And we've got ERG11 drawn here. It's on all of the amplifications here. And we're just assuming that that's the driver. It could be that there are other genes in this segment that are driving it, which has of course also been found for pathogens, that multiple linked genes can contribute to these resistance traits. Another one that we saw over and over again was amplification of a segment of chromosome 15. And we think the driver of this is amplification of the drug pump PDR5, which is also a known antifungal resistance mutation. And then we saw a few that included this gene, UPC2. This one we're a little less certain about. As you can tell, one of the amplifications doesn't contain this gene, so it's got something else going on. But UPC2 was a good candidate because it's kind of a master regulator of ergosterol biosynthesis. And so we think that amplification of this is a good candidate for this, but it's really fun to have the students kind of go through these in they can look up the different genes on SGD, Saccharomyces genome database, and try to figure out, what genes might have this trait, be associated with this trait, and could multiple genes in a segment, and then they get to start thinking about how cancer genomes evolve, which often have lots of copy number changes and things like that. We saw lots of mutations in the genes PDR1 and PDR3. So these are paralogs. They encode a transcription factor that regulates a whole bunch of the drug pumps and other things like that. And nearly all the strains had a mutation in one of these. And we think that these are gain of function mutations based on information in the literature about what types of mutations these are. And we actually also did some evolution experiments with diploids and still got these. So we think that is also supportive of that. This is just a subset of the mutations that we got, but mapped onto the structures or the gene models of PDR1 and PDR3. So there are all sorts of different mutations that came up. And this is one of those cases where we probably haven't saturated the space. We see a couple of mutations twice, and we see some same sites, but to different mutations. And so this is fun for the students because when they get their list of mutations back and they're trying to figure out, which mutations matter, they start typing them in, PDR1, pleiotropic drug resistance 1, and they're like, oh my God, it worked, right? So they then can say to the other groups, what mutations do you have? And a pattern emerges once they start collecting and comparing that, this is a very common mutation that they got. Petites are also really common, and this was another case. This is something that I didn't know before we started this, but it is known that losing respiratory capacity does cause drug resistance. And so I love these pictures because they look like those, I don't know if any of you ever ate those candy dots on paper as a kid, but they look kind of like candy dots to me. And so here are our ancestors, and then there's evolved strains pinned out after them. And this actually shows one of the other interesting things about this experiment that we have not really nailed down yet, but you can see there's some color variation among the evolved strains. And the pigments are expressed off of high copy number 2 micron plasmid, but we do see some modulation and copy number of that and occasional loss, even though we are selecting for the plasmid. But anyway, so you can see all the ancestors are capable of growing on glycerol, non-fermentable carbon source, but many of the evolved strains are not. Interestingly, we have some teams where like most of their strains were able to grow and others where none of them were. And so we haven't quite figured out like what the patterns are that might explain some of that. But we found that these petites were really common. This is also a really interesting trait for the students to think about with respect to trade-offs. Because here's a strain clearly superior growing in a drug condition, but now it's lost this capability to grow in this other condition. And the students in particular are faced with this when they do competition experiments where they mix 2 cultures together and then plate them to count colonies. And they see that the petites are out competing a less fit strain, but yet their colonies are small. And they're like, but how can it be winning and yet make smaller colonies? And so then they have to really wrestle with like, what is fitness anyway? What are we selecting for? And they kind of come to some of their own conclusions about that. Okay, like I said, we have so many replicates now. This is probably, there's one selection pressure in my lab we've done more replicates of, but not by much. So we were able to do enough replicates here that we're starting to see patterns emerge that we typically haven't been able to really be confident of in some of our lower powered experimental evolution work that my lab has done previously. And so this has been a really exciting aspect of this, and one of the parts where having so many classrooms working on the same problem together has actually been really beneficial. We were having conversations about, should we do more azole resistance? We're learning the same targets over and over again? But this shows some of the value in even doing more replicates of something that is reasonably well understood. So this is just a snapshot. This is not by any means all of the mutations. This is just some of the Westridge high mutations from 2 years, and both the early and final time points for the ones where we have those. And if you just look at PDR1 and PDR3, for example, you can see that nearly all the strains have one of these, but none of the strains have mutations in both. So this is a case where these are mutually exclusive mutations, and we made the double mutation and tested its growth, and it at a kind of a medium drug concentration, the double mutant does not in fact have an advantage versus either of the single mutants. So we think that there's no benefit to getting a mutation in that second locus. We are now doing a few new experiments to really do more careful looking at different concentrations of drug and see is there a drug level where it does become beneficial. And it does look like it's possible that the double mutant has a higher minimum inhibitory concentration, but we are still in the process of finishing those experiments. So I decided not to show the data for that just yet. Another pattern that we noticed was this, and you kind of sort of see it a little bit over here, but so here's ERG25. So ERG25, we often see decreased function mutations, and we never see stop codons in this, so we don't think it's total loss of function. But it's known that ERG25 auxotrophs stop the buildup of certain intermediates in the ergosterol biosynthesis pathway, which are toxic. And so this could be a way of reducing the toxicity and creating resistance in that way. And we found these other mutations in 2 transcription factors, HAP1 and ROX1. And these are both heme-containing transcription factors. They sense oxygen and they regulate just a ton of stuff in the cell, including some of ergosterol biosynthesis. And so interestingly, the lab strain that we use already has a mutation that disables HAP1, but we saw additional point mutations that introduced stop codons. So we think that further disabling of HAP1 is beneficial to these strains. We're not entirely sure why, but we got this over and over and over again, right? And so what we noticed was that we very rarely saw a HAP1 or a ROX1 mutation without there also being an ERG25 mutation there. And so here is just the tally up of everything. So we have about half of our strains got an ERG25 mutation, but nearly all of the mutations in HAP1 or ROX1 occurred in the context of ERG25 mutation. And we saw a similar but less extreme pattern for ATP1 and ATP2, which are mitochondrial genes involved in mitochondrial function, and these also seem to occur mostly in the context of an ERG25 background mutation. So we think that by doing so many replicates of this, we can start to see some of these patterns. And we're now in the process of making these double mutants so that we can actually compare their phenotypes and ask, is a HAP1 mutation more beneficial in the context of an ERG25 mutation versus on its own? And does that explain some of this type of patterning that we're seeing? Oops. Okay, so we've looked at like I said, 99 strains. It's over 500 mutations across all these strains, and we focused on ones that occurred more than 3 times independently. We've done a kind of a half-baked simulation just to say, like, how common would it be to get the same gene multiple times just by chance? And 3 seem to be the threshold for unlikely by chance. So we have a bunch of mutations that meet that threshold, and many of them are known to be involved in azole resistance. So we're pretty confident that they're likely to be causative. But also we find some new mutations in genes where the pathway is known to be involved in azole resistance, but that particular gene we were unable to find evidence for in the literature of a specific mutation that caused antifungal resistance. So that's kind of exciting that we're now kind of getting more saturation to the screen. And some of the things that come up in a statistically significant way are mRNA degradation and P-bodies, sphingolipid production, which makes a lot of sense, and the RAM signaling network, which is kind of interesting and might be about modulating growth in response to these insults. We were also excited about this category, which is a novel thing. So, that was one of the things that we really mentioned to the students, was the possibility that they might discover something new. And that it happened has just been great. So CSF1 is our favorite example of this. So CSF1 is an uncharacterized mitochondrial protein. It's not really clear what it does or what other phenotypes it might have, but we found it mutated 5 times in our data set. So what Bryce did was he went back and he recreated this mutation alone by itself in the wild type ancestor strain using CRISPR. And the way that he did that creates a synonymous mutation that gets rid of the PAM site. And so he built a control strain that only had that synonymous mutation and a strain that creates also the mutation that's similar to one of the ones that we found in our evolution data set. He then mixed these strains together and competed them. And he competed them either in rich media that didn't contain any drug or in the media that contained the FungiCure. And he used, again, kind of a medium dosage of this that the ancestor could grow in, but where it is not very good at it. And so after doing several rounds of serial dilution, mimicking the experiments done by the students, in this case, we didn't have these marked with different colors. So he used the sequence itself as kind of a poor man's barcode here. So all we do is we Sanger sequence this little chunk of the gene, and you can actually see on the electropherogram where there are 2 bases for the 2 different nucleotides that are present in the pool. And you can measure the heights of those and watch the changes over time and get a not bad estimate of the selection coefficient. And so from these data, he then turned that into a frequency. And so what happens is at the beginning, the strain that contains the mutation is present at just a portion of the population. If you compete them in YPD, that doesn't change much. But if you compete them in FungiCure, the CSF1 mutation quickly zooms up and takes over. So that's really nice. We were able to confirm one of these novel mutations that we found. What it has to do with azole resistance, we're still not entirely clear about, but because it's in the mitochondria, that leads to some hypotheses about how that it might be doing something similar to some of these other mitochondrial mutations that we're seeing. Okay, so how do we get the students involved in this? So like I mentioned, they are doing searches in Saccharomyces genome database. They also look up things in the literature. They're pretty good at internet research, it turns out. And this has made me a little more, I should say, enthusiastic about publishing open access and putting things on bioRxiv, because a high school student searching for your paper, you want them to be able to read it, right? Like, so they were able to check out ideas about different genes that they find, and they did a writing exercise about their favorite gene and why they think it might be related to drug resistance and what experiments they could do to test that. And they also were able to present their work. So Ryan does a lot of work with the students to make posters that they can go and present at research conferences, for example. Some of the students came and visited my lab and did a little presentation, which was great. And this one, they're actually presenting at an undergraduate research conference. So the high school students are getting to kind of go and see what other students at colleges are able to do. And you can see, they're talking about PDR1, PDR3, the chromosome 8 duplication, and the petites, all in this poster here. And one of my favorite things is a story that Ryan told me where the students had like a free day where they could work on whatever they wanted to, some sort of independent project. And several of his students chose to spend hours in his lab Googling yeast gene names to try to learn more about their evolved strains. And one of them at the end said, I feel like this is what a real scientist does. And I mean, gosh, you just can't beat that, right? This has just been really great fun to get to tap into that enthusiasm that the students have. They also just get attached. So this is an email that Ryan shared with me where one of his students wanted to wish him good luck for a presentation that he was giving. And they call them their yeast babies and their yeast families, and are just really, really into it. And one of the other reasons that they're really into it is this competition angle. So I mentioned this a couple of times, but I want to talk about this as it's kind of like the capstone of the teaching lab. So once they have their evolved strains, they can mix them and compete them and use colony counting to tell which strain is the winner. We've actually tried a few times to see if we can measure the relative frequency by looking at the color of the liquid culture. I had an undergraduate who was trying to build us a gadget that would do this. And so far, we unfortunately haven't had much luck, because it turns out plating for single colonies is pretty tough. So not all the experiments are the best for this kind of data collection, but at least usually you can figure out who the winner is. Even if the plating leaves something to be desired. And so we do this like bracket style. We set up a whole yeast madness bracket, and the students get super into it and who's going to win. And they know a little bit about their mutations at this point, right? And so they're like, oh, my mutation, I've got a PDR1 mutation in here that I think is going to, take it all the way this year. And one of my teachers in Seattle, she involves the entire school in this. So even though it's her biology classrooms that are doing the competitions, she lets anybody fill out a bracket. And so the students are all like, how's your yeast doing today? Did your yeast make it to the next round? And they get really into it, and they learn a little bit about, what kinds of mutations does the winner have? And they, it's hard to back out like who's winning because of those mutations or anything, but the students do appreciate this kind of aspect of, who has evolved the best yeast. Okay, I want to end just by mentioning that we also have a big assessment wing of this. So we wanted to make sure that first we were doing no harm, that we weren't hurting the students' performance on things like their AP exam, and that we were improving things like how well do they understand evolution? Are they more or less interested in pursuing science careers? Do they feel more or less confident about doing experiments themselves? And so we did a series of surveys, and also enlisted Alexa Warwick, who is an education researcher, and she devised a whole coding system for this, and has really brought a lot of professionalism to what was just kind of talking to the teachers and students and asking questions beforehand. So it's been really great to work with her. And stay tuned, we should have a preprint about the pedagogy aspects of this up on bioRxiv in the next couple of weeks. And so I'm not going to go through her results carefully, but I just want to say that we were really pleased to see that their knowledge of how they think about evolution improved, and also was more nuanced. They showed improved interest in pursuing science careers. They showed improved confidence that they could design a scientific experiment and carry it out. And Ryan in particular was very happy to see that their AP test scores did not suffer. So the administration was happy about that aspect. Okay, so the experiments that I've told you about were all with this antifungal, but of course you can use anything, right? Anything that yeast can eat or that inhibits the yeast is a substrate that you could potentially do this experiment on, right? So one of the ones that we started doing thanks to an inquiry from a biotechnology classroom is wood waste. So at the University of Washington, we've got this great forestry department. And so a collaborator there has been supplying me with hydrolysis from chips, bark, and branches. So essentially trash from the wood industry, and to see if, and yeast can eat the sugars in this, but there are some sugars that they don't prefer that's in this, and there are also a bunch of inhibitors. And so we've evolved yeast in this condition now and seen some really neat mutations that they get. We've also done caffeine. This is the condition we chose for our pandemic project, which was trying to evolve things at home. And I wanted something that would be absolutely safe in case, you know, somebody's kid sister drank their experiment or something. Another fun one has been invasive growth. So this was the idea of one of my undergraduates, Margot Walson. So Margot thought, you know, what if we could come up with something that was a little more compelling as a phenotype, like a visual phenotype. Like the students can tell that their strains are evolving resistance because they're tolerating higher and higher concentrations. But what if it was something you could see more easily? And so she became enamored of this really cool trait that mostly wild yeast have, where this is, you know, if you make just a puddle of yeast on the surface, they'll grow into this kind of big colony. But if you wash those off, then you can see this residual kind of shadow here. And that's actually yeast that have grown into the plate. And this invasive growth phenotype is, of course, really important for pathogenicity. And so what she has been doing is taking this chunk here. She pulls out a chunk and uses that to inoculate the next round. And after just a few passages of this, she was able to evolve more invasive yeast. And so we're trying to modify this to make it a little more contamination proof, let's just say, but I think that this has a lot of promise as another way of doing evolution in the classroom. We've done more strains. One thing we're really interested in is industrial strains, in particular with this wood waste experiment. And this evolving at home kit that we developed, I'm really excited about the prospects for that. You know, it came out of necessity because none of our students were in the classroom anymore where they could do their evolution experiments. But these are the kits that Bryce prepared for my lab to do the beta testing. Here's me. I had orange yeast and evolved it in the sludge experiments. That's what we call the wood waste hydrolysis. It's the sludge evolutions. And so these involve a bunch of pre-filled test tubes that have snap caps so they don't evaporate, a packet of the individually wrapped sterile cotton swabs, and a tea candle so they can create their own little Bunsen burner, and a little notebook so that they can record their observations. And this actually, we're hoping will have life beyond the pandemic, because this might allow us to work with more high schools to have some students maybe do a summer project to bring the evolved strains back to the classroom in the fall, and other potential applications. And then, you know, finally, you know, getting more labs in on this. This is something where you can really tailor the conditions to your own interests, and you can work with your own local schools. And so we're kind of using a franchise model where we can help get you set up with strains, protocols, learning materials, scaffolding for the teachers, and then you can create your own little YEvo franchise at home. So if you'd like to learn more about this, we've got a website, we've got a Twitter feed, and like I mentioned, we've got our preprint out about these mutations, and I'd love it if any of you read it. It'd just be fabulous to get some feedback on that, particularly from the point of view of some of these mutations and how they work at a little higher level of understanding than maybe what we've been able to put together as newcomers to this field. And we'll have a new preprint in the next few weeks on the pedagogy aspects. And with that, I'll just end and thank all the people. So Bryce is right here, and he was really the leader of this whole charge, and it's just been fabulous. And I'm really excited for what his lab is going to work on at Loras. And I've tried to acknowledge our other collaborators as I went. And we've been really lucky to get NSF funding for part of this. So I'm going to stop sharing now so that I can see you all while you're asking questions.
All right, thank you very much, Maitreya. That's awesome. It's so inspiring to see like people get excited about science and wanted to do more than what they were asked. I think that's great. Yeah.